Hi everyone, my name is Fatemeh, and today I'm going to talk about something that has the potential to change the future of type 1 diabetes treatment. But before we get into that, let me tell you a little bit about myself. So, as I mentioned, my name is Fatemeh. For this project, I worked with my mentor Sophie, who helped guide me through my research, and lastly, I'm based in Vancouver, Canada. Okay, first, I think it's important for us to understand what type 1 diabetes is. So, type 1 diabetes is an autoimmune disease, which means that the body's own immune system mistakenly attacks and destroys healthy cells. In this case, it specifically targets the beta cells in the pancreas, which are responsible for producing insulin, a hormone that regulates blood sugar levels. To understand why this is a problem, let's look at how things work in someone without diabetes. As you can see in the diagram, normally, when we eat, carbohydrates are broken down into glucose, which enter the bloodstream. In response, the beta cells in the pancreas release insulin, which acts like a key that unlocks cells so they can absorb glucose and use it for energy. This keeps blood sugar levels stable. But if you look on the right, in someone with type 1 diabetes, the immune system mistakes these beta cells that make insulin for harmful invaders and destroys them. And without beta cells, the pancreas stops producing insulin entirely. Without insulin, glucose cannot enter cells and instead builds up in the bloodstream, leading to dangerously high blood sugar levels. Now, why did I choose to focus on this disease? Well, because it's a huge.
Oh, sorry, quick interruption. My Zoom is still there on your slide. If you just click it out of the way, I just would hate
Oh,
for it to
oh
block
yeah,
the
yeah,
slide anymore. Perfect.
yes. Thank you.
Yeah.
Okay, so why did I choose to focus on this disease? Well, because it's a huge global issue, yet it still has no cure. Here's what the numbers tell us. 9.4 million people worldwide, including children, are living with type 1 diabetes. Every year, over 81 billion is spent on managing the disease. And despite medical advancements, 175,000 people still die from it each year. But these numbers only tell part of the story, because living with type 1 diabetes is incredibly hard. The current treatments, like insulin injection and pumps, are expensive, time consuming, and don't fix the root problem. So the big question is, can we do better? That's where stem cell therapy comes in. So let's explore why stem cells are such a powerful solution. First, stem cells can self renew. Second, and most importantly, they can differentiate, meaning they can become many different types of specialized cells, like muscle, nerve, or insulin producing beta cells. If you look at the diagram, you'll see this in action. A single stem cell divides and then transforms into a completely new cell type. This ability to become other cells is unique to stem cells and is what makes them so valuable in regenerative medicine. In type 1 diabetes, this means we could replace the lost beta cells and restore the body's ability to make insulin, going beyond just managing the disease to actually repairing it. Now, not all stem cells are the same. So let's start by looking at the first type, pluripotent stem cells. So, pluripotent stem cells are one of the most promising tools in regenerative medicine. And this is because they can differentiate into cells from all three GAMS layers, such as the ectoderm layer, which is shown in yellow, the mesoderm layer, which is shown in red, and lastly, the endoderm layer, shown in blue, which develops into internal organs, including the pancreas, where beta cells are found. And since beta cells come from the endoderm, pluripotent stem cells are ideal for diabetes treatment because if we can guide them to become beta cells, we might be able to restore insulin production. There are two main types of pluripotent stem cells. There's embryonic stem cells and induced pluripotent stem cells, and each have their own advantage and limitation, which I'll go over next. So, starting with embryonic stem cells, these are derived from early stage embryos, specifically from a structure called the blastocyte, which forms about 5 to 7 days after fertilization. And if you look at the diagram, we can break down this process step by step. So, it all starts with a fertilized egg. And after fertilization, the single cell zygote begins to divide, forming a small cluster of cells. This cluster develops into a blastocyte, which is a hollow ball of cells that contain two key components, the outer layer and the inner cell mass, which contain pluripotent stem cells. Scientists extract these stem cells from the inner cell mass and grow them in the lab, where they can be directed to become specialized cells. And embryonic stem cells are powerful because they are naturally pluripotent, but using them involves destroying an embryo, which raises ethical concerns. And that's why researchers have also turned to alternatives, like induced pluripotent stem cells, or iPSCs. So, unlike embryonic stem cells, which are derived from embryos, iPSCs are created by reprogramming adult somatic cells back into a pluripotent state. And if you look at the diagram, we can break down the process step by step. So first, we must isolate somatic cells and culture them in a dish. Then scientists introduce a set of specific transcription factors, which reset the cell's identity, allowing it to return to a pluripotent state. And then over a few weeks, the cells gradually lose their specialized identity and transform into fully pluripotent stem cells. And once pluripotent, these stem cells can be directed to differentiate into various specialized cell types, as shown in the diagram. And iPSCs don't require embryos, so they eliminate the ethical concerns of embryonic stem cells, and they also reduce the risk of immune rejection as they can be generated from an individual own cell. But they still have their own challenges. For instance, their programming process isn't always perfect. Some cells may carry genetic issues or like remember their old identity, which can affect how well they work. Also, one of the reprogramming factors used is linked to cancer, so there's a risk of tumor formation if it's not carefully controlled. So while iPSCs are promising and ethically flexible, we still need to make sure they're safe and reliable for clinical use. So once pluripotent stem cells are obtained, either as embryonic stem cells or iPSCs, the next challenge is directing them to become fully functional insulin-producing beta cells. In the human body, this happens naturally during fetal development. But in the lab, we need to mimic this process step by step using the right signals at the right time. And if we mess up even one step, the cells might become something else entirely. So let's walk through the key stages using the diagram. So we start by activating two key signaling pathways, activin or nodal and WNT. These signals push the cells toward the endoderm lineage, which, as we discussed, gives rise to organs like the pancreas. But we're not done yet. The endoderm also gives rise to other organs, including the lung and liver. So next, we add signals like FGFs and retinoic acid while blocking others like SHH and BMP to guide the cells specifically towards the pancreas. At this point, we have pancreatic cells, but the pancreas is composed of multiple cell types. So we now need to specify an endocrine cell fate, which is the lineage responsible for hormone production. And we do this by using EGF and T3 and continue blocking unwanted pathways like NOTCH and CHH. And finally, more signals like EGF and BMP are used to refine the cells into ones that can actually sense glucose and release insulin in response, like natural beta cells. And as you can see at the bottom of the diagram, throughout this process, these signals trigger the activation of important transcription factors, which are proteins that turn specific genes on or off. And each transcription factor plays a role in moving the cell to the next stage of development, guiding it step by step from a generic stem cell to a specialized beta cell. By the end, the cells express a set of key transcription factors that allow them to produce insulin, sense glucose, and function like a real beta cell in the body. And even though we can now create beta-like cells in the lab, there are still challenges as many of the lab-grown beta cells still don't respond to glucose as well as natural ones. But there's still a lot of hope. The process keeps improving and researchers are getting closer every day to making this a real and effective treatment. So now that we covered how stem cells can be used to generate beta cells, let's shift our focus to another type of stem cell that plays a very different role in treating type 1 diabetes, hematopoietic stem cells, or HSCs. So unlike pluripotent stem cells, HSCs are multipotent, meaning they can differentiate into a limited number of specialized cell types within a specific tissue or lineage, which in the case of HSCs is blood and immune cells. And if you look at the diagram, we can see HSCs live in the bone marrow, specifically in the red bone marrow. Since HSCs create the immune cells, such as white blood cells, scientists are exploring them as a way to reset or rebuild the immune system so it stops attacking the beta cells. Let's look at how this therapy works. So, unlike using stem cells to replace beta cells, this approach targets the underlying autoimmune problem by resetting or modifying the immune system. And this process is called hematopoietic stem cell transplantation. And as the diagram shows, it involves multiple carefully controlled steps. So let's break it down one by one. So step one is mobilizing HSCs from the bone marrow into the bloodstream. And to do this, doctors administer pretreatment medication, which stimulate the bone marrow to release HSCs into circulation. Step two is collecting the HSCs from the bloodstream. Step three is freezing the collected stem cells, while step four is being completed because step four takes a while. Step four is immune system reset using chemotherapy. So before reintroducing the patient's own HSCs, patients undergo chemotherapy, which partially or fully removes their faulty immune system, which is necessary because it clears space in the bone marrow and removes the faulty immune system. And this step, this step comes with a lot of risk because it temporarily weakens the immune system, making the patient more vulnerable to infections. Step five is transplanting the HSCs back into the patient. So once the immune system has been removed, the stored HSCs are infused back into the patient through a simple IV infusion. And these stem cells can travel to the bone marrow where they begin repopulating the immune system with new healthy immune cells. And lastly, step six, recovery and immune rebuilding. So, overall, this therapy has shown promising results in some clinical trials, with some patients experiencing a reduction in insulin dependence and even periods of restored insulin production. So, to wrap up, we've explored how both pluripotent and hematopoietic stem cells offer exciting possibilities for treating type 1 diabetes, from replacing lost beta cells to retraining the immune system. And while there are still challenges like cell function, survival, and safety, ongoing research and improvements are bringing us closer to a real long term solution. Thank you so much for listening.
Yay! Thank you so much for sharing.
Yeah,
And you can go ahead and stop sharing your screen
okay,
too.
perfect.
Awesome. Um, I don't know what your current career aspirations are, but I think you would be such a good high school science teacher. I was just sitting here like, this is like a better explanation than I ever got like in high school or even undergrad. So thank you so much. I feel like I learned so much too. I didn't know there were different kinds of bone marrow. This was such a cool presentation.
Thank you.
I'm curious, what like first made you interested in learning more about type 1 diabetes and looking for more effective solutions?
I mean, I have like family members who have type 1 diabetes, and also my dad and like my mom, they do a lot of research with type 2 diabetes. So I thought, let me be a little bit different and choose,
Yeah.
but still be on the same path. So that's
Yeah. Oh, I
yeah.
love that. I love that. What was kind of the most challenging part of your research and going through all of this material?
Well, I think it was definitely turning the pluripotent stem cells into beta cells. I think that process was really hard. Like it's so confusing to understand, like how the signaling pathways are used, like how that turns the transcription factors on, the whole thing. But Sophie helped me a lot. But yeah, it took a while to understand that.
Amazing. Amazing. Would have taken me even longer, so great job.
Yeah.